beginning part of this lecture looking at chapter 3. We were looking at the cells, and we looked at the plasma membrane and the cytosol and the different organelles that make up the cell and what their functions are and things like that. And then we saw a little video, and now I told you that we, in this particular lecture set of notes, we skip into chapter 6, and we're going to talk a little bit about transport across membranes. We'll talk about membrane potentials here and cell signaling, just kind of in general introducing these concepts to you. And then I haven't posted yet, but I will. We're going to go back to chapter 3. There's chapter 3 part 2 and chapter 3 part 3, which I'm going to be posting, and that's going to take us through mitosis, meiosis, and gene expression, which is protein synthesis, how you make proteins from genes. So we'll talk about that as well, and then we'll be getting into metabolism. So let's go ahead and, like I said, when you're studying this, just make sure that you pay attention to the figure labels that are on the figures in these notes so that you know which chapter to go to. So in this part of chapter 6, we're going to look at the extracellular environment briefly. We'll talk about diffusion, osmosis, carrier-mediated transport, which includes things like facilitated diffusion and active transport. We'll talk about membrane potential, and then a little bit about cell signaling. And again, this is sort of introducing these concepts. So some of this stuff, again, is a basic concept that we will be discussing later in application to an organ system that we're going to be describing processes for. So first of all, the outside environment of the cell, known as the extracellular environment, includes all, really a lot of water. But most of the body water that we have is within the cell itself. And when you think about that, that makes a lot of sense, right? We know the cytosol, it's this jelly-like material, but it consists mainly of water. It consists of a lot of water. And we've got billions of cells. We have epithelial cells, we have nerve cells, we have muscle cells, we have, you think all 3 types of muscle cells. You think about all the cells we have tons and tons of cells. So most of the body water is inside the cells, known as the intracellular compartment. 33% of body water is outside the cells in the extracellular compartment, basically the tissue fluid. 20% of this water, the 33% that's in the extracellular compartment, 20% of that is in the plasma of the blood, but 80% is in the interstitial fluid, which is the tissue fluid. So we talked about connective tissues. Connective tissues are always going to be deep to epithelial tissues, and we know that connective tissues have a matrix, right, which consists of a specialized cell that secretes this matrix, which consists of protein fibers and ground substance, right? Ground substance is going to consist of a lot of water, and that is what we're talking about. So intracellular compartment has most of the body's water. The extracellular compartment has 33%. Of that 33%, 20% is in the plasma, 80% is in the matrix. Okay, so this is what basically it looks like. So again, most of the body's water is found in the cells. Here we've got some epithelial cells. Here's some fibroblasts. We've got blood cells, which also have a cytoplasm, and so forth and so on. Now, all of this around the blood vessel is connective tissue. And again, in the connective tissue fluid, we have a great deal of the water in the extracellular compartment. And then we have a little bit in the plasma of the blood. Now, some other things that are found in the outside of the cells, the extracellular matrix, We have collagen and elastic fibers, and we also have reticular fibers, right, that provide a kind of a structure and framework. But they are linked to molecules of gel-like ground substance and to plasma membrane integrins. So generally speaking, the cells have a link. So like this is a fibroblast, right? Bless you. This is a fibroblast which secretes this matrix out here, the protein, fibers, and ground substance. How does it know what protein fibers that it has to produce? How does it know this? It knows this because there is an integrin protein that will connect this cell basically to the outside environment, and it can communicate, and it will know what kinds of proteins it needs to produce. It's kind of an interesting network that is there. The glycoproteins link intracellular and extracellular compartments. Those are the integrins. And then the interstitial fluid resides in hydrated gel of the ground substance. Again, that's what we were talking about. This is the ground substance of the connective tissue. That's where you're going to have the water. So like in areolar connective tissue, for example, that's sort of jelly-like, that's where the water would be. Do you have any questions about that? That's it for the extracellular compartment. Now we're going to talk about movement across the plasma membrane. And I think once we get finished with this section, we'll take a break. But first of all, we know that the plasma membrane is made up of what? What is the macromolecule that primarily makes up the plasma membrane? Yes, the phospholipid, right? That is the canvas for our fluid mosaic, right? So phospholipids make it up, and it's very important because we need to be able to regulate or control what gets into the cell. I mean, obviously the plasma membrane gives the cell its shape, but we need to be able to also regulate what gets into the cell and what leaves the cell. So the phospholipid bilayer, the plasma membrane, is selectively permeable. It only allows certain kinds of molecules to pass, and only at certain times. Many important molecules have transporters and channels. So for example, glucose is a pretty big molecule. C6H12O6, big molecule. In order to get into a cell, it needs a transporter to be able to do that. If the cell didn't have transporters for it, it couldn't get in there. Also channels, things that are charged like sodium or chloride, for example. Those things are charged. They're going to need some type of a channel to get themselves from one side to the other. In general, we can say that transportation across the cell membrane can either require a carrier or not. So carrier-mediated transport involves a specific protein transportation system, whether it be a channel or some type of a transport protein. Non-carrier mediated means that a molecule can just get through the plasma membrane by itself, and that occurs by way of diffusion, which we're going to be talking about in just a second. So that's a very general classification about transportation. Does it require a carrier or doesn't it? A little bit more specifically, we can define transportation by talking about energy requirements. If we have a situation known as passive transport, that means we require no energy. Basically, things go from an area of high concentration to low concentration, and they can pass directly through the plasma membrane. They do not have to use a channel, a transporter, anything like that. So passive transport, again, requires no energy at all. It's very much like if you are in an elevator that has 20 people in it, right? Think about the elevator down the hall if there were 20 people in there. You would be so tightly packed, right? As soon as that door opened, it would take no energy for you at all to want to get out of there, right? You would go from an area of high concentration to low concentration. You would just go right out into that hallway, and you would feel a lot better, right? That's basically what passive transport is. Active transport, on the other hand, requires energy. It moves substances against the gradient, requiring energy in transportation. So again, I always use this example. If, say, somebody's coming to your house and it's a mess, pretend like you have like a bunch of clothes in the middle of your floor or junk, right? And you're like, gotta get rid of this really quick. I need to take it out of the floor and I need to put it someplace where I can hide it. So you open your closet door and there's already a ton of stuff in there, right? And you pick up your clothes and you pick up your junk that's in the floor and you shove it into the closet against the gradient, right? You've already got high concentration of stuff in that closet and you're trying to put more in there. And when you go to shut the door, like, you gotta really push it, right? It takes a lot of energy to get it into that area because there's already so much stuff in that closet already. That's basically active transport. It takes a lot of energy to move things against a gradient from an area of lower concentration in your room, for example, to an area of higher concentration in your closet. So that is the energy requirement classification. So do you have any questions about that so far? Okay, so we have passive transport. We're going to start talking about that. We have diffusion. Diffusion is the simplest method of transportation. It requires no energy at all. It is random motion of molecules, basically from an area of high concentration to low, and these molecules can pass through a membrane. Now this is basically sort of this diagram is showing us kind of like an experimental design. If we had a membrane that we put into a container of water, and this membrane was artificially designed to be, permeable to small things, we could put a couple of drops of dye into one side of the membrane, and what would happen is over time, because the dye is highly concentrated on one side, it's going to diffuse to an area of low concentration. The net diffusion they show here with the thicker arrow is going to be from the side of the higher concentration to lower concentration. But there will be some diffusion back across the membrane as well. And this will continue basically until we reach equilibrium. In other words, when the dye molecules are the same amount of dye molecules are on this side of the membrane as they are on that side of the membrane. At that point in time, the diffusion will stop. Because what drives diffusion is the movement from high concentration to low. And things are small enough they can pass directly through the membrane. So this is a physiological example. These are tissue cells, and this is the extracellular environment. Our blood vessel would go through here, drop off some oxygen, which is shown by the blue circles here. And then the yellow circles are carbon dioxide, which is made by the cells. So when the oxygen is dropped off by the blood vessels, the oxygen being in high concentration outside in the extracellular environment is going to diffuse with the gradient directly across the plasma membrane into the cell. Whereas the carbon dioxide being in high concentration inside the cell is going to diffuse out following its concentration gradient and then into the bloodstream, which isn't shown here. Does that make sense? Oxygen, carbon dioxide, they're gases, but they're very small molecules and they can pass through the membrane directly through simple diffusion. So anything that's nonpolar can diffuse through the cell membrane. Some small molecules like carbon dioxide and oxygen can go through. And also, and I know this seems counterintuitive, but water in, for example, one molecule of water is very, very small. And the phospholipid bilayer, although it's lipid and it repels water, there are gaps in between the tails of the phospholipids, and one individual water molecule could actually make its way through and in between the phospholipids in the plasma membrane. Not a whole bunch, but like maybe one little molecule could, because it's very small. Yeah, so pretty much anything that is nonpolar, anything like or a gas or like a really small molecule of water can get through the membrane through simple diffusion. Now, sometimes, like if we had a charged particle like sodium or chloride or potassium, you know, any of those types of things, they have a charge, and that's a problem with the plasma membrane because the plasma membrane is going to repel any type of charge. So sometimes what we need to have channels that allow for diffusion to occur. So that would be like in a way, a carrier-mediated sort of transport. What we talked about previously was simple diffusion, where things can just pass directly through the membrane. Here, we're still talking about diffusion, but we're talking about needing a channel in order to facilitate that diffusion dynamic, because a molecule has a charge and it's not going to be able to just go right through the phospholipid bilayer. It needs a channel. And sometimes, and it's interesting, most cells do have channels that are always open to certain ions. And we're going to be talking about that in the nervous system. Sometimes the channels are just, they're open, they're always open. So cell membranes are impermeable, again, to charged and most polar compounds. Charged molecules have to have a channel or a transporter to move across the membrane. So again, we're still moving from high concentration to low. So if this is, higher concentration inside here, or the extracellular fluid, and it's lower inside the cell, we need a channel to get these things through. But it's still going from high concentration to low, does not require energy, just requires diffusion. In terms of diffusion, some of the variables that affect how quickly diffusion can occur, aka the rate of diffusion of a compound, depends on, first of all, the magnitude of the concentration gradient. Obviously, if you had 5 people in the elevator versus 20, you're going to want to get out of that elevator with the 20 people a lot faster than you would want to get out of an elevator with 5 people, right? So essentially, the more you have in terms of, the more of a particular component you have on one side of the membrane versus the other, the faster the diffusion rate will occur. The permeability of the membrane to it. Obviously, if we have more channels for a particular thing, or the membrane is just open to this particular molecule being able to pass through it, then the rate of diffusion would be a lot faster. The temperature. Now, temperature is a tricky thing, and when we see temperature listed in other areas of this course, temperature, you have to remember, as you increase the temperature, it does increase pretty much all activity. When you increase the temperature, the rate of diffusion will go up. But can you continually just raise the temperature and continue to, no. Eventually it's going to plateau and then drop off. Why is that? What class of macromolecule? What class of macromolecule would carriers be, or channels? What would they be? We got 4 macromolecules to choose from. I'm going to sit down and wait until what are our 4 macromolecules that we talked about? Lipids. Our channels, do you think they're lipids?
Yes. Or carbohydrates.
Okay. Okay. Carbohydrates is another macromolecule. Do you think channels would be carbohydrates or lipids?
Probably proteins.
You're right.
Yeah.
Very good. Proteins, right? You know, not nucleic acids because that's DNA and RNA, but proteins. These channels, these carriers, a lot of times they're proteins. So can we just continually increase the temperature, and then how quickly we diffuse is just going to keep getting faster and faster? Because what happens when we add too much heat to a protein? Don't you remember the example I gave you of the egg? What happens?
Changes. It stops being a protein.
It denatures. It doesn't stop being a protein. It just becomes a dysfunctional protein. It is going to denature, which means it unfolds and it won't be functional anymore. It can't refold again. So that's what happens, denaturation. Okay, so yeah, temperature, as it increases, it will increase the rate of diffusion, but only to a certain point, after which case the denaturation occurs, and that's it. And then the surface area of the membrane. Obviously, the more surface area we have for a particular membrane, the more we can have diffusion occurring, right? But it's also going to depend on how permeable the membrane is to it. I mean, we can have a lot of surface area, but if we don't have a lot of channels, or we don't have a lot of amenability to being able to have that molecule diffuse, then that's not going to matter, right? So surface area of the membrane is kind of dependent upon the permeability of the membrane as well. But all of these have a directly proportional relationship to the rate of diffusion. In other words, as we increase the magnitude, the permeability, the temperature, the surface area, we increase that, we increase how quickly diffusion occurs. But again, with temperature, that only happens to a certain point, after which time denaturation occurs and no diffusion will occur. All right, so far we've talked about passive transport, we've talked about simple diffusion, where substances move from high concentration to low across the membrane, and basically doesn't require any energy, doesn't require any carriers. Then we talked about diffusion that did require a carrier. What kinds of molecules can pass through using diffusion, high to low concentration, not requiring energy. What kinds of molecules need to use a channel to diffuse? Charged particles, and anything else? Things that are polar. Very good. Good. All right. So now we're going to talk about osmosis. And osmosis also doesn't require energy, but it's different than diffusion. Okay, because with diffusion we require a concentration gradient. We move from high concentration to low. Osmosis is different. Osmosis also, in a way, well, it doesn't require energy, but it does require something. It requires what's known as an osmotic gradient, okay, or a solute gradient. Now, what are solutes? Solutes are things like sodium, chloride, glucose, so basically sugars, ions, proteins, all that kind of stuff is a solute. A solvent is water, and we talk about this. Now, osmosis specifically is the movement of water, and again, it requires a solute gradient. So let me ask you this. If you ate a bag of potato chips right now, what would you want to have near you? Or some pretzels? Something to drink, right? And preferably water, because if you drink something else that has sugar in it, you're still going to feel thirsty, right? Because water follows what we call solute. So solvent, water, always follows solute, which is the salt and the sugar and so forth. So keep that in mind as we talk about this. Okay, so first of all, Osmosis is the net diffusion of water across a selectively permeable membrane. Water diffuses down its solute gradients. Now, this thing that looks like Swiss cheese here, does really look like Swiss cheese. This is a membrane. This is supposed to be a membrane. And the blue circles are water, and the pink large circles are solutes. Now, in order for osmosis to work, in order to draw water to one side of the membrane or the other, our solutes have to be too big to be able to pass through the membrane, because if our solute goes back and forth, the water is just going to pass back and forth, and it's not going to at all provide any kind of pressure to draw water just to one side. So we call this osmotically active. In other words, these are solutes which cannot freely move across the membrane. They're too big to pass through themselves. So I don't know if you remember in lab, but we were talking about the capillaries, and I mentioned that there are proteins in the blood, in the capillaries, and on the venule side of the capillary bed, the water tends to get pulled back in to the capillary, and then I said the rest of the water that doesn't is going to go into the lymphatics and then be carried back to the circulation. Do you remember that in the lab? Anybody? Okay. Well, okay, I know you don't. But it's the same idea. The proteins in the plasma proteins, they're too big to get out of the capillaries, so they're going to exhibit a pressure to draw the water back into the capillary bed, and we call that osmotic pressure. So you'll hear that term. So here you can see we have, on this side of the membrane, we have 4 solutes. This side we have 2. So notice that the net movement of the water molecules is going to be to the side where we have more solutes. Make sense? Do you have any questions about that? So these solutes have to be osmotically active. They can't cross through the membrane in order to exert a pressure. In this case, these solutes are exerting a pressure that draws water to the right side. Okay, so that's pretty much the principle of osmosis. So water diffuses down its solute gradient until its concentration is equal on both sides of the membrane, unless we're talking about something like the capillaries I just described. Venule side, you want to draw as much water back in as you possibly can. Some cells have water channels that facilitate osmosis. These are known as aquaporins. So some cells, when we were talking earlier about simple diffusion, sometimes water molecules can get through the membrane directly, passing through, but sometimes they use these little pores called aquaporins that are like little water channels. They're little pores that allow the water to come through. Okay, so osmotic pressure, and this is what I was mentioning earlier, is going to be determined by how strong your osmotic pressure is determined by how much solute concentration, what your solute concentration is on one side of the membrane. The more solute you have on one side of the membrane, the more strongly water will diffuse. It's a pressure. It's kind of like if you took a little pinch of salt, you put it on your tongue, or you took a tablespoon of salt and you put it on your tongue. Which one's going to make you the thirstiest, right? The tablespoon, right? I mean, it's going to be, so the more solute you have, the more strongly water wants to go there. All right, so that's osmotic pressure. Now we're going to talk about tonicity, and this has a clinical relevance. Tonicity is the effect of the solution on osmotic movement of water. So essentially, when we talk about tonicity, we are talking about the solute concentration of a solution that a cell is actually in. So there are three types of solutions that we can have. We can have a solution that is hypotonic, So I'm going to draw this. This is a beaker of water, and there's a cell, okay? And our solution, it's hypotonic, which means it's below. So the solute concentration in our solution is less than what's inside the cell. So let's draw Na here, that's our solute. One outside, and then we'll draw two inside, okay? So now, the question is, what direction will the water most likely go? Is it gonna go into the cell, or is it gonna leave the cell? Okay, you think it's gonna leave it. Why do you think it's gonna leave it?
You said water.
Yeah, water. So we have one sodium in the fluid surrounding the cell. We have two sodiums inside the cell. Okay.
It's more concentrated inside the cell.
Right, so you told me that water is gonna leave it, right? Why do you think the water would leave? Any idea? Huh?
What I just said. That's not the answer.
Oh, okay. Anybody have a different answer?
Just stretching.
Oh, you don't want to raise your hand, you just want to start. All right. Okay, remember, water goes to the side of the membrane where the concentration of the solute is the highest, right? Where is the concentration the highest? Inside or outside the cell?
Inside the cell.
Inside the cell. So what direction will the water then go? Into the cell or out? In, exactly. So in this case, water moves in. Now, if too much water comes into the cell, what can happen to it? If you're adding water to the cell, it's going to wilt. It'll burst. Actually, a cell could burst. See, plants, okay, I don't know. Do you guys know anything about, like, flowers? You plant flowers in the summer, you know? No? Okay. Well, you ever see, like, you ever see an aloe vera plant, or, like, I don't know, impatiens? Those are, like, those red or white flowers that they like to grow in the shape.
Succulents?
Yeah, or
My
succulent.
drowned.
What?
My succulent, like, it got too much water from being outside.
Okay,
Like, looks like it's gonna
burst.
explode, yeah.
Okay, but it doesn't, and the reason plants don't burst like that is because they have a cell wall. They have like a double membrane in their cells, so they don't burst. So when you add water, okay, because you know, you think a plant, the plant cells are going to have a lot of glucose and things like that. So when you add water, that's just going to puff, you know, it's going to go in the cells, they're going to puff up. And that's called turgor pressure, actually. Our cells, we don't have a cell wall, we just have a flimsy plasma membrane. So if this was a red blood cell, for example, this would rupture if we had too much, So we call that actually hemolysis. There's that lysis suffix again that I told you guys about, like we talked about hydrolysis, right, reactions, which was on the quiz today. Hemolysis would occur, which is a rupture of the red blood cell. Okay, so now let's do the opposite. If we have a hypertonic solution, that means we have more solute outside the cell than we do inside. So what direction does the water now go? It's going to leave, right? It's going to go out. So the water leaves the cell, and now what could happen to the cell? previous answer we gave?
Sure.
Yeah, they'll shrivel, right? It could it could shrivel up. And again, if this was a red blood cell, we would call this crenation. Crenation could occur. Okay, so last situation we can have is if we had a cell that has the same solute concentration as that which is in the fluid surrounding it, we would call this isotonic. Iso means the same. Iso is the same. So it's got the same tonicity. The solution has the same tonicity or the same solute concentration as that which is in the cell. What's the net movement of water going to be?
Neutral.
Neutral. There's no movement. No movement, right. So I'm going to give you an example here. Now, if you had a person go into the hospital for dehydration and they would give a person saline solution, it's usually going to be 0.9% saline if you look at the bag. Okay, so 0.9%, I can tell you, is isotonic to your red blood cells. What if they gave a person a 1% solution? Would that be hyper or hypotonic?
Hyper.
It would be hypertonic. And what could happen then? Would, huh? Well, if it's hypertonic, that means that there's going to be more solute where? In the cell or out? Outside. So what's going to happen? The net movement of water is going to be what? In or out? Out. So what could happen to the cell? It would crenate, right? They would shrivel. Likewise, if we had a solution that was, 0.5%, that's going to be what? Hypotonic, right? If 0.9% is isotonic, 0.5% would be hypotonic. So, yeah.
Wouldn't it be higher than 9?
Say that again.
Wouldn't it be higher than 0.9 to be hypertonic? Don't you want more sodium than 0.9 to make it hypertonic?
Remember tonicity refers to the solution that's surrounding the cell. So if it's hypertonic, the solution has more solute than what's inside the cell. So when the previous example I gave, if it was a 1% solution, then that's hypertonic. If it's 0.5%, then that's hypotonic, because it's less than 0.9.
Oh.
See, if we're saying 0.9% saline is isotonic to the red blood cell and that solution is isotonic, that means that the solution has the same solute concentration as that which is inside the cell, okay? If we go to a 0.5% solution, that means that we have less solute in that solution than we have inside the cell. So in that case, what direction does the water move? Into the cell or out?
Into the cell.
Into the cell. The solution is hypotonic. It has less in the solution than what's inside the cell. So the water goes into the cell. Right, and that can cause what?
Bursting of the cell.
Right, hemolysis, rupturing of the cell. Exactly. Good. So remember, that's one thing when we're talking about the tonicity, we're describing the concentration of solute in the solution that surrounds the cell. If it's hypotonic, it's less in the solution than what's in the cell. If it's hypertonic, it's more in the solution than that which is inside the cell. If it's isotonic, then the solution and the cell have the same concentration. Okay? So, let's recap just for a second. So far, we've talked about diffusion, right? Simple diffusion doesn't require any type of channel. But then we do have ions and things that might need a channel, but they can still diffuse. How does diffusion occur? What does it require? It requires a what? Well, it's a form of transport. Does it require energy? Never. It does not require energy. But it does require what? The elevator example.
Osmotic pressure.
You know, diffusion. Okay, so
Passive transport.
no. Diffusion requires a concentration gradient. It requires movement from high concentration to low concentration. Diffusion does, okay? It can occur simply without a transporter or carrier, or it might need a channel like we talked about. Now we talked about osmosis. Osmosis is specifically the movement of water, okay? Does it require a concentration gradient? No, but it does require a solute gradient. And the solute gradient is going to draw the water to one side of the membrane or the other. Does this make sense to you? Do you have any questions about this? Because you know what, as simple as these concepts may seem, they do trip people up sometimes. It tripped me up when I was a student. Not real sure why, but it did. Okay, so I really don't even know why I need the screen. This whiteboard does just a fine job, right? It does a fine job. Okay, so this is just showing us what we just talked about. If the cell's in an isotonic solution, the red blood cells it stays the same. If we are in a hypotonic solution, that means that the solute concentration outside the cell is less than what's inside the cell. So water moves in and it could cause this rupturing of the red blood cell. If it's hypertonic, that means that there's more solute outside the cell than what's inside. So water is drawn out and that can cause the cell to shrivel or cremate. Okay. Any questions about this so far? All right.
I have a
So,
question.
yes.
Is to compare osmosis and diffusion, are they like two sides of the same coin, or are they more like opposites of each other? Like,
Both.
what's their relation to each other?
Well, osmosis is just the movement of water. Diffusion could be the movement of oxygen, carbon dioxide, ions, polar molecules, lots of different things. But osmosis is specifically the type of transport that occurs for water.
So water can't diffuse. It can only osmose.
One water molecule could, but like a bunch of them could not. They would have to go by way of osmosis. So both of those types of transport are passive. They do not require energy, but they require a gradient. Okay, diffusion requires the concentration gradient. Substances moving from high concentration to low. Osmosis requires a solute gradient, moving to the side where the concentration is the highest. Okay? Any other questions? All right, so regulation of blood osmolality is what we're going to talk about. Osmolality refers to solute concentration within the blood, in the plasma. So this is a negative feedback loop. If you dehydrate yourself, then obviously your plasma volume is going to go down because your water is found in your plasma. So you drop your blood volume, which is, of course, if you dehydrate, you're going to increase the solute concentration, right? Because you don't have as much water to dilute it. So you dehydrate, you drop your volume of blood, you increase your osmolality in your plasma. This is detected by receptors in the hypothalamus, and the hypothalamus does make you feel thirsty, when you need water. Osmoreceptors in the hypothalamus pick this up, and two effects occur. One, you feel thirsty, you drink more, and you increase your water intake. In addition, you also get antidiuretic hormone, ADH, antidiuretic hormone secretion from the posterior pituitary. And this is what I was saying. The posterior pituitary, can always remember, prevents pee-pee. antidiuretic hormone prevents diuresis, which is urination, okay? So in other words, what it's going to do is cause the kidneys to retain your water and bring it back into the bloodstream. So those two effects together, increasing your water intake and having the kidneys increase that retention of water in the blood and not urinating it out, is going to increase your blood volume, which will dilute the solutes in there, and it drops the plasma osmolality, and that's a negative feedback loop. So again, you dehydrate yourself, you decrease your blood volume and increase the osmolality. Osmoreceptors in the hypothalamus pick this up. Hypothalamus makes you feel thirsty, you drink more water. In addition to that, it triggers the posterior pituitary to secrete antidiuretic hormone, which prevents PP, posterior pituitary, and it eventually will cause the kidneys to increase the water retention. You don't urinate it out, and the blood volume is going to go up, the osmolality drops. And that's it. Negative feedback. All right, so next we have carrier-mediated transport. So this last type of transport we're going to talk about here is also passive, but it does require a membrane protein carrier. So these are for molecules that are too large, like glucose or polar. They can't just simply diffuse. They can't just make it through a channel. They need an actual transport protein to get them through. So I'm going to skip this slide for a second, and I'm going to just show you facilitated diffusion. Okay, so anytime you see the word diffusion attached to transport, that means that energy is not needed. Energy is not needed. Okay, but you need a concentration gradient. Okay, from high to low concentration. Now, the term facilitated means that we need a carrier protein to facilitate or to get the diffusion process going, because these molecules are too big. So, for example, this is glucose, which is very large, and it needs a carrier. It's in high concentration outside the cell, low concentration inside, so the glucose molecule will bind to its transporter. When glucose binds, that causes the transporter to change its configuration so that it opens up to the inside of the cell so the glucose can be released. This doesn't require energy, but it does require the concentration gradient. Glucose here is moving from high to low concentration, and it does require a transport protein. Okay? With me? Almost time for a break, promise. Okay, I'm gonna go and just explain this. Yeah, we'll wait. I think we'll take a break after the diffusion stuff, and we come back. We'll talk about active transport. But let me just talk about this slide first, and then we'll take 10 minutes. Okay, so in terms of protein carriers, like what we just saw, the glucose transporter, they exhibit a couple of characteristics. They exhibit specificity for a single molecule. Generally they do. Sometimes they can transport more than one type of molecule. It really depends. But also we exhibit competition among substrates for transport. With carriers that can bind to two different types of molecules, there will be competition. And there's saturation. When all the carriers are occupied, this is called the transport maximum. So I'm going to illustrate this for you a little bit. If we had a bus that had 20 seats, and we had at the bus stop, we had 20, let's see, we have 20 men, 20 men. If we had 20 men at the bus stop, and that bus came every hour, how many men would we transport per hour? 20, right. What if we had 25 men at that bus stop? How many men would we transport per hour? Okay, I'll recap. The bus has 20 seats. There are 25 men standing at the bus stop. How many, the bus comes every hour, how many men would we transport per hour? 20. Doesn't matter if we had 20 men, 10, if we have 10 men there, it's gonna transport 10 men, okay? What if we had men and women at that bus stop?
Still 20?
It would still be 20, but now there's competition. And it's not gonna just be men that are transported, it's gonna be women too. So this is what this diagram is illustrating here. How quickly transportation of a particular substance X, how quickly it occurs. If X is just by itself, like the men at the bus stop, you can see that you can more quickly transport X. But once we reach a point where all of the carriers are saturated, then we plateau. Just like with the bus, whenever 20 people were there, right? You can only transport 20 people per hour. But now if we have competition where there's X, but there's also Y that wants to be transported as well, we reach our saturation point more quickly and X can't be transported as fast. It's pretty much what this diagram is showing us. So it's pretty intuitive and logical. It's not very complicated, but sometimes when people see these kind of diagrams, they think it's a lot more tricky than it is. Do you have any questions about diffusion, simple diffusion, facilitated diffusion, or osmosis? So again, one little recap, then we'll take a break, come back and talk about active transport. So to reiterate, we have simple diffusion does not require a channel. Anything that's small or nonpolar can pass through this way. Then we have diffusion that does require a channel for things that are charged and things that are polar. They can pass that way. But simple diffusion is passive transport. It's transport that is passive. It doesn't require any energy. It requires movement from high concentration to low. Likewise, facilitated diffusion that we just talked about also requires substances moving from high concentration to low, but it requires an actual transporter. This is for things that are too large, like glucose. Osmosis doesn't require energy either, but it requires a solute gradient, and water will follow, go to the side where the solute concentration is the highest in the membrane, right? None of these require energy, but they do require a gradient of some type. And in the case of facilitated diffusion and diffusion that requires a channel, we need some type of carrier to help us. Make sense? Any questions? All right, go ahead, take 10 minutes and come back about 10:42, and we'll talk about active transport. Yeah. Is it good? It is? Oh, okay, cool. I'll check it out.
It looks better, and it does everything you explained.